NASA's Jet Propulsion Laboratory presents the Von Karman Lecture, a series of talks by scientists and engineers who are exploring our planet, our solar system, and all that lies beyond.
Good evening, everybody. My name is Marc Razze from JPL's Office of Communications and Education, and it's my pleasure to be your host, welcome you, and thank you for joining us for this September edition of the Von Karman series. Our co-host today is my friend and colleague, also from the Office of Communications and Education, Miss Nikki Wyrick. Hello, my friend.
Hello. Thank you so much for having me tonight. I'm super excited to be here and I'll be handling all of your questions this evening. So please do remember this is your space program and we want you to be involved in the conversation. As you're watching, ask questions in the chat box and our social media team will bring in as many as we can to the talk tonight. Keep in mind, if you don't see the chat, make sure you refresh and it should be right there for you.
Thank you very much, Nikki. Appreciate that. So let's jump right in, shall we? So now, many of you may know that one of NASA's goals is to search for life in our solar system and beyond. But that's no small task. We found places in our solar system where we believe that conditions exist that could potentially allow life to take hold, but they're not nearby. And of course, the question, what exactly is life, can further complicate the issue. But we've got to start somewhere, right? Tonight, we're going to be talking with Dr. Marc Wronkiewicz, a data scientist at JPL who currently leads a team building autonomy software to detect and characterize cell-size life for the Ocean Worlds Life Surveyor, or OWLS project. Hey, Marc, thanks for joining us tonight, and hey, happy birthday.
Thanks. Hey, Marc and Nikki, good to be with you guys.
We're thankful, really grateful that you could join us tonight. So before we get too deep into this, tell us a little bit about yourself. How did you get to where you are?
Sure. So I'm originally from the Midwest. I went to school at WashU in St. Louis doing engineering, and then after that, I did neuroscience at University of Washington in Seattle. And I like to say, kind of towards the end of my PhD, I got just as interested in artificial neural networks as the biological ones. I kind of got caught up in the machine learning and that data science craze that was really taking off in the mid 20 teens. So after I graduated, I went over to a small company in DC called Development Seed, and worked there as a machine learning engineer. We got to work on very fun projects like working on social good issues, so working with the World Bank, Red Cross, those type of organizations. But after that, I kind of felt this, I guess I would call it like a calling almost. I don't know. I felt this feeling that I wanted to get more involved in exploring some of the other places within the solar system. So I hopped over to JPL 2 weeks before lockdown. So I got to come in, meet a few of my coworkers, and go straight back home. And then a couple weeks after that, I got involved on the OWLS project.
Well, you picked a difficult one, I'd say. Looking for life is clearly challenging, right? So let's go here and ask, what do you think it would mean if we actually found life?
Yeah, I think, even we're just looking for, just looking for microscopic life. I think it's hard to overstate and maybe even imagine how all the different ways that would affect us if we were to find life off Earth. So, I mean, even just from the scientific perspective, think of like biology, chemistry, genetics, like all these fields would be upset overnight. There'd be, have a brand new thing to look at. It'd be huge for all those fields. But I mean, even society at large, you think about like in the 15s and 1600s, Copernicus and Galileo, they're doing all this work and saying like, hey, so it looks like in our data that actually the universe does not revolve literally around Earth. And those were like huge controversial statements. And, Galileo actually faced like the court of the Inquisition for pushing ideas like that. So that was just to figure out like what was our spatial point in the universe that we sit at, right? So if we were to discover we're actually also not alone in this universe and we found life, we're looking within our own solar system, that would be huge. That would really change a lot of how we conceive of ourselves in the universe.
It would be an amazing find, to say the least, I think. So where are the places we're looking, and I guess what makes that challenging?
Yeah. So we're kind of taking the strategy of follow the water. All life that we know of depends on liquid water to support its chemistry. So there are a few places actually in our solar system where we can find that. So yeah, this is a perfect image here. Uh, this is from the Cassini probe that was out exploring Saturn and some of its moons. I think this picture came back in 2015. Don't quote me on that. But when this came back, this was like a huge holy cow moment for that science team. So what you're looking at here is actually water geysers spraying out into space from this moon Enceladus. And there's actually, you can see a number of different geysers. So there's these huge cracks in the south pole of Enceladus, and it's spraying this water out into space. And with a little bit more analysis, what the scientists who are working on this figured out is that there are liquid oceans beneath this outer icy shell of Enceladus. And so that makes this a pretty interesting and good target for us to go look for things like microscopic life, if we want to go and search the water there. One of the challenging things, there's a bunch, and we'll get to them. One of the challenging things is that Enceladus is very far away from Earth. So, like Saturn itself can be up to 10 times further away from Earth than the Sun is. And basically, it is very difficult to communicate over those distances just because of the rules of physics. And I will touch a little bit more on that later. But if you think about exploring some of these places, your ability to transmit information from them back home to Earth is really, really hard.
So let's step back a little bit, I think, and ask, like, well, do we know? Do we have a definition for life? And if we do, like, then how do we continue to look for it?
Yeah, that's another one of the big challenges. It's almost paradoxical because we're looking for life. We don't have a good definition for it. I mean, you can find one in a dictionary, but basically, there is no good set of like check boxes, if you will, that you can go down and say like, all right, if I see these like 12 or 15, whatever number of conditions, and I check all those boxes, then I found life, right? That doesn't exist. And basically, it's because anytime you try to write down a definition of life, every time you write some criteria, you end up with 10 exceptions that go with it. So it becomes this futile exercise to try and figure out what exactly you're looking for. So the OWLS project is it's built in a way upon that conundrum, if you will. That is a bit of a cornerstone of the project. And so the way we approach that then is say, let's look for life in as many different ways as we can. So we're interested in looking at microscopic organisms. So, we'll get into this, but we have a whole bunch of instruments kind of on the chemistry side that are looking for very small scale things, all the way up to the biology side that are actually looking for individual cells. And the idea is you get as many different instruments looking for different signs of life as you can. And so we call those, like, we're looking for as many independent biosignatures as we can. And the hope is that if there is something there, that you see it across a number of different instruments that are all looking in different ways, and that's how you get to a firm and like scientifically defensible conclusion that we have indeed found life if we come across it.
Okay, that makes sense. So let me ask about autonomy then. That's a big part of this project, right? So maybe for those
Yeah,
folks who aren't necessarily familiar with that term, can maybe define autonomy for us?
sure. Yeah. So very broadly, autonomy just means the ability to make your own decisions. And so for us, we are specifically developing onboard science autonomy. And so I'll break that down. So we have autonomy, and in this case, it's software. It's going to be looking at data and making decisions based on that data. It is on board. It means it actually lives on board the spacecraft, so it's running on board like the flight computers. If we send a lander to one of these places, maybe actually we can pull up slide 2 here, and I'll kind of talk through these ideas there. So we're on board, we're autonomy, and we're science focused. So that's what the science autonomy bit is, meaning we're building this software to try and we would say maximize the scientific return of the mission. So basically, that just means, in this image here, we have our lander kind of on the left, and it's pulling. We feed in our water sample, and we're going to feed that water sample to a bunch of different instruments, like we talked about. The autonomy is going to look at all that data on board and try to pull out, like, what are the best nuggets, what are the best biosignatures we see in this data and pull those out and then send just those little bits back to our scientists. Because remember earlier we talked about how it's really, really difficult to transmit data back from these faraway locations. So that's kind of the, stepping back. That's what this whole autonomy, the science autonomy we're building is trying to do. It's trying to empower our scientists. Given that we can transmit such little data, we need to make sure we transmit the best data we've found back to them to help them make those firm scientific conclusions about whether or not we see life.
So tell us a little bit more then about those actual complications. You mentioned distance, and what else like makes this so challenging?
Yeah, absolutely. So the distance piece is really fun. So if you look at how much data that we can collect, our very best case scenario, we can transmit something like 1/1000th, 0.1% of the data back to Earth.
Wow.
That is like taking the entire Hitchhiker's Guide to the Galaxy and condensing it down into a single tweet. And so you gotta capture as much of the literary content as possible, the story arc, the humor, all the characters, all of this in a single tweet. That's our best case scenario. We're probably going to be looking at something more like 1/10,000th of the information. So like 0.01% of the data we collect will be able to transmit back. So that's the conditions we need to make sure that we enable our science team back here on Earth to be able to make firm conclusions about whether or not they see signs of life. So the autonomy itself, it kind of has three different components to it. I'll very just briefly go over. So when the data comes in, the first thing we'll do is a bunch of sanity checks essentially on the data. We call this like a data quality estimate. And it's just looking to make sure there's nothing off about the data. And if there is something that we don't want to spend valuable transmission bandwidth on that data. After it goes through those checks, we go through this summarization step. And so for each of the instruments, the software is looking at this data and trying to extract out what are the important biosignatures that we might see in that data. If it's there, it needs to extract them and compress them. And we work with the scientists quite a bit to get all those algorithms honed. After we've done that for all the instruments, then the final step is to take all of our now summarized science data and prioritize it. So it kind of like creates this big long queue from top to bottom of what is the highest priority data, make sure we send that back first, and then kind of like work backwards through all that data and send that back to our team.
You probably had to work with a pretty enormous team of folks then, I would guess. What was that like? How many people were you dealing with at the time?
Yeah, this was probably one of the most rewarding parts of the project. The team was very big. So there's 3 or 4 dozen people that worked on the project. Everything from biologists to chemists to very talented instrument developers on both the biology and the chemistry side, and as well as us on the autonomy team. And we also worked with the flight hardware and software teams, so the people that are actually building the hardware that will run, control these instruments and then allow us from the ground to communicate and work with the lander. So it was a huge team, and we had to talk to everybody. It was a great position to be in because we needed to take input and work with all those different groups. So on the science side, I think it's hopefully pretty obvious, right? Like the scientists are going to are looking at this data. So they're telling us what's the most important components of their data. And we need to make sure that we're building software that can find and extract those biosignatures, those signs of life. So that was, weekly meetings, right? We're going back, hey, we worked on adding this new algorithm. Here are some of the results on the data you have. What do you think? Like, oh, that's good, but actually we got this new data set in last week that's very different. So we're going to need you to go through this again. And then, okay, we found out this doesn't work on this specific use case. All right, we'll get back to you next week and just kind of repeatedly do that. So that was a really important piece because that's, we're building up that relationship and that trust with the science team to make sure that we can find and extract what they're most interested in. In a way that's also really scary because you are standing between the scientists and their data, which is not something you usually have to deal with on Earth, right? If I tell you as a scientist, you
Yeah.
can only pick out 0.1 to 0.01% of your data, and someone else's software is going to do it. That's going to be really scary for a lot of people.
Yeah, no doubt.
So
You
yeah, a lot of
develop
work with the scientists. Oh, go ahead. Yeah.
I was going to say you must develop quite a trusting bond with those teams, right?
Yeah, and I think that was absolutely vital. And that was something that was really special about the project. Like a lot of times in these research projects, you don't always get to spend years working with a science team from us on like the autonomous software perspective. So that was something that was very special that we got to build up that bond over a long time. Um, and then, even working with the instrument developers as well, like we need to understand a lot of the ins and outs about the raw data that's coming off those instruments. I mentioned earlier that we've built up this battery of, sanity checks for the data. That actually becomes really helpful to sit next to the instrument developers and say like, oh yeah, you collected, 4 hours of data yesterday. I can run through and analyze that in, maybe 20 minutes. And I have all these checks that will run and flag things if there's any problems with the data. So as they're developing the instrument, adding new things, that becomes kind of nice to be able to sit alongside them and help them in some ways. I mean, they didn't really need the help. They're also very talented, but it's a nice way to provide some value. And then on the hardware side too, I think that another one of the big challenges of doing this um, is that you have to make it run on a flight computer. We don't have server racks. We don't have cloud computing. We don't have any like nice high performance computing software or computer or anything like that. We're running on something that's kind of like a modern smartphone. Like that's as much as we've got. So working with them to say, okay, we have to make sure that all the algorithms we develop also run in a reasonable amount of time. They can't take 2 days to run when we can collect many samples a day.
So I know we've got a bunch of great slides. So, and I know most of them relate to this. How do you test this thing here on Earth?
Yeah. So maybe we can pull up slide 8 to start with, just to show you guys what did it actually look like. Yeah, so we took our instrument suite out to the field, out to Mono Lake, and we'll get to that in a second. But this is actually the OWLS platform itself. So 3 boxes on the left. That's where all the flight computers live. In the middle is a bunch of the different chemistry instruments. So there's a mass spec and several other chemistry focused instruments. And then on the right side there, we have the microscopes live in that box. So we took this out to Mono Lake, California. It's just a little east of Yosemite, kind of close to the Nevada border. And there, Mono Lake is actually, in some ways, a pretty good analog for what we would see on Enceladus. I mean, it's not like frozen over anything. We're in desert California, but the lake is hypersaline. It kind of lies at the bottom of this old volcano caldera, so all the water is always running down into the lake and it never leaves. So it gets very salty. And that's what we expect to see actually on Enceladus. So we can go through like slides 9 through 13, just kind of like slowly flip through those and show you what Mono Lake looked like. Yeah, so that's kind of like looking out from the parking lot into the lake itself. So we went there to stress test this entire system. We, spent a lot of time developing it in the laboratory and working on all these different components. But at the end of the day, we needed to go out to a field site, show that we can take up samples of water, um, introduce them to all the different instruments. All those instruments are collecting data, looking for those biosignatures, all those different biosignatures we talked about, all those independent lines of evidence. And once that data is collected, then at the end of every day, we would come in and run the autonomy software on that data and try to and confirm that we could extract the biosignatures and kind of give a report back to the team each morning. So it's hard to describe how much fun it was to go there and work. We were there for like 6 days. And it was basically like a huge hackathon where we're making sure we can, go end to end on the system, um, debugging problems along the way. But at the end of the day, with, all the team's really hard work, I think we had like about 20 people that went. With all the team's hard work, we were able to show success on all the instruments, and we found actually a couple of motile swimmers in the lake itself. So a couple of cells that are actually swimming around on their own, which was really cool because we didn't know if we would find that. There was a lot of algae and things there too, so that that sign of life was very abundant, maybe more than we'll find on Enceladus. Um, but maybe I can show a couple examples of like what the instrument data we're actually looking at
Yeah,
looks
what's that
like.
data look like?
So Yeah. I think on slide, or so I'll just quickly describe two of the instruments, and then we can show some data from them. Um, the first one, one of the really cool microscopes on the project is called this digital holographic microscope, or DHM. And when I first saw this, I was like, holy cow, this is way more interesting and sophisticated than like microscopes I dealt with in biology 101. All right, instead of having like a slide, like preparing a slide and looking at it through a microscope, this microscope can image a 3D volume. So we basically flow water, a small, like through a channel past the microscope's field of view. And that microscope can image essentially what's like a microscopic swimming pool. So it's like a small, about a millimeter by a millimeter, I think by 1/2 millimeter deep um, volume. And what that means is, this microscope, the reason it's there, the biosignature we're looking for, is motility. So we have this essentially what's a microscopic swimming pool. If there are any cells that can move on their own, they can swim around and move around in that volume. And we can capture that on the microscope. So maybe we can show slide 3 here that has some videos of the raw data. Yeah. So what you're seeing here is you see a bunch of particles kind of drifting down from the top right to the bottom left. And they're just kind of passively moving through. Those could be like dead cells or debris. But you can also see some of these cells are kind of corkscrewing and swirling around on the screen. So that those cells are swimming, they're motile. And this is one of the key biosignatures we're going to look for on places like Enceladus. Because if a cell or something like a cell can move on its own, that is really evolutionarily advantageous, because that means you can go swim and find food and also do a better job avoiding becoming somebody's food. So on top of this, what you're seeing is these tracks being drawn on those particles, and that's actually the autonomy at work there. So it is tracking some of those more visually salient particles, and those are what those lines are. And then on top of those tracks, then we have some machine learning algorithms that analyze those tracks and try to determine whether or not they're motile. So they're taking in various track features, like how fast was it moving, how often did it turn, things like this, and making a prediction about whether or not they think those cells are alive and moving. And so that's one of the key biosignatures that our science autonomy is trying to extract. So instead, at the end of the day, instead of having this like, I think it's about 5 gigabytes per minute of raw data, because this is 3D data. It comes in basically in 4K, so it's generating a bunch of information. The autonomy takes that, extracts these tracks, and extracts little, almost like particle portraits, little raw cutouts around any of the particles it sees, as well as some other contextual information. And that's what's transmitted back to the science team. So you end up going from something like 5 gigabytes per minute. You can compress that down to maybe about 5 megabytes. So that's how we get our compression here. So this was on the biology side, but we also worked with the chemistry team. So maybe I'll describe the instrument in a second, then we can flip over to that video. On the chemistry side, what we're looking for is very small molecules that may indicate life. So things like, think amino acids, right? Building blocks for proteins. So on that side of the house, basically what happens is we pull in our water sample. It basically goes through like a mini pressure cooker. So if there are any cells in that specific sample, they'll get cooked and kind of burst open, and you end up with what's essentially almost like cell soup that then you can feed to things like this mass spectrometer. And that kind of exposes, if there are things like amino acids, they're now exposed in this fluid. So maybe if we can go over to slide 5, that's going to show some of this data here. Yeah, so we get this mass spectrometer. It's looking for peaks in the data. So this would be when these things like amino acids are split up, right, as they get fed into the mass spectrometer, they will show up as little peaks in this raw data. And so what the autonomy is doing here is saying, I'm going to go through and search and find where are all those peaks in this data, identify them, make these little cutouts. Again, you can kind of see that pattern. It's making these little cutouts of the raw peaks, and then pulling together some different statistics about how high they are, how wide they are, things like this. with this information, then the scientists can go through and use these peaks almost like a chemical fingerprint. So I actually saw them do this in the field. It's really impressive. You can say, all right, I saw a peak at 150 m over z at uh, like 22 minutes, and there's another one lined up at this mass, and they'll be like, okay, that sounds like it could be guanine, without even like looking at anything. It's really impressive. So this is with this data here, then they can go back and say, okay, are there any of these like organic molecules within the data that would indicate signs of life, like amino acids or nucleic acids, things like that. Um, yeah, hopefully that makes some sense about how we're taking the raw data, extracting these key signatures, and transmitting just the most important pieces back to our science team.
That's pretty amazing. It's got to take, without that power you've got to deal with, it's got to be so extremely challenging to narrow down the software to be able to do that. So kudos to you and the team for sure. Um, how long does it take to build something like this instrument package that you've had out there at Mono Lake, for example?
Yeah, it is. So this is a long time in the making. I think, the specific OWLS team came together and worked for um, like a little over 3 years on this, on integrating all these different instruments. But even the individual instruments, right? Those same teams were working on those well before OWLS even started. So it takes a long time. It takes a lot of dedicated effort, and it takes a lot of collaboration. I think that's kind of the real key takeaway is having the team together working as one, kind of like rowing all in the same direction on this really tough problem. Um, that was what I think helped us be able to put together this entire integrated system and and be successful in the field. I think maybe it'd be good to show maybe like slide 14 and 15, a couple more images from our field test. Yeah, so here's Aaron, Mauro, and Nate. They're putting together, kind of setting up the system underneath the tent. We were out basically in this parking lot outside the visitor center, set up there for like 6 days. So they're just getting things set up now, and right after this, I think they kind of set up the command central where all of the operations were run from, just on the right, on one of these like picnic tables. And on that, I think on the next image, yeah, this is uh, this is Jess and Seth, and these these two were kind of in a way like heroes of the project, especially one of the days. They were getting up at like 5 a.m., maybe even earlier each day, going out on Mono Lake on a boat and pulling up water samples so that every single day we'd have a fresh sample to run through all the instruments and run all the autonomy on. And I think maybe it was the last day they were collecting samples. Actually, the water, it started getting really windy out. And the reason they would go early in the morning is to avoid the wind and avoid getting any whitecaps on the water. And one of the days it did pick up. And so they had to ditch the boat and hike back. I think it was like over 2 miles in the heat with the water samples, and they were wearing like full body waders. And they had to hike all the way back to camp because it was too dangerous to be out on the water. So I think that just speaks to the dedication of the team. Everyone was really working hard because we really believe in what we're doing, and it was, again, I can't capture the energy that was there in the field to work alongside all these great people.
You're doing a pretty good job. You can just kind of, we're vibing it from you for sure. Had to be like the coolest science camping trip ever, right?
Yeah,
So
I think
I guess a
we have
question.
one team photo
Oh, go
to
ahead.
go. Maybe go one
Yeah,
more
go ahead.
photo forward, and we can show the whole. Um, this is just the team that went out to the field. Unfortunately, we didn't have the money to bring everyone out there because it was, as you can imagine, like a week-long field campaign is pretty took a lot of resources. But yeah, maybe flip forward back and forth between this photo and the next one. Yeah, so it
Love
was a
it.
really, really fun group to be working with. Very smart people, but just so much fun. I don't know if I've had a more rewarding like single week of work in my life. It was great.
That's just great. That makes me so smile. So then what has to happen now before owls can become like a real life flight project?
Yeah. So I th- doing this field test, the whole point of this is to push this as far along towards a real mission as we can. So that's why we went out there. That's why we stress tested the system, put it through all its paces, tried to identify any places where we can improve, but also showing that we could rely on some technology that's already out there. So, for example, things like we used F Prime, which is a open source flight software package used to control like probes we send out in the solar system. That's actually used right now to control Mars helicopter Ingenuity. We also, as I mentioned at the start, we use this very light compute platform that's really similar to the Snapdragon platform that runs in a lot of mobile phones. That is also in use right now by Ingenuity, the Mars helicopter. And so the goal here was to try and show we can rely already on a lot of technology that's already proven, and that makes the path towards getting things like OWLS on a real mission and actually get sent out to Enceladus. That really shortens that path. So at the end of the day, what we were trying to do is really show there's no miracles that need to happen for OWLS to get it ready for flight. We try to push it as far as we can and understand the limits of the system and get it to the point where we can start actually thinking about proposing this on some of the future missions we want to send out to ocean worlds out in the outer solar system.
So yeah, following that along, then, what kind of future missions do you see for this type of autonomy?
Yeah, uh, it is a really interesting and fun area that's it's kind of blowing up right now because data has become so cheap and easy to collect. Um, you think about now that we get a global image of the entire Earth every day. We're NASA's working right now on the NISAR orbiter, which is going to it basically images and kind of collects data about the 3D surface of Earth. That when that gets launched, that will produce. Don't quote me on this, but I think it's more data per year than NASA has ever collected on all of its missions over all time. Like, it's crazy. So the amount of data that we're gonna have, it just keeps exponentially rising, and so having things like science autonomy to help us focus our attention and understand what is how do I understand where the interesting bits of my data are? How do I put those pieces of data in front of scientists and humans rather than having to hire, armies of grad students and interns to manually flip through images or, like, look through data? That's just not gonna be tenable anymore. So that's one piece of it. We wanna take all those same, um, instruments and orbiters and things that we can use right now here on Earth, because we're so close, we're able to transmit the data pretty easily back down to the ground. But if we could take all those same fantastic scientific instruments and send them out to Mars or the asteroid belt or further out, like we're trying to go to Enceladus, you're going to run into that problem that we talked about at the start, that physics just says that you are not going to be able to transmit very much data. It's worse than dial-up out there, and you need something like this onboard science autonomy to be able to extract out the best, most scientifically interesting components of your data, however the scientists define that, and transmit that back to Earth. So that's it's very exciting. Like one of the very exciting ways this is going to help enable more science, and as we explore the solar system. But even here on the ground, there are so many areas where we could help save scientists time by just pointing them to the most interesting bits of the data. So think back to those microscopy videos I was showing you, where we're trying to find and track anything that seems to swim under its own power. That can be useful in a medical setting, for example. If you're looking at blood from someone who may have sepsis and you want to see if there's any bacteria swimming around in their blood, and there's all these red blood cells, and it's really hard to track just the one or two bacteria you're looking for, rather than having a person manually look in a microscope and kind of like be turning the knob and focusing back and forth, seeing if they see anything, send like a larger blood sample through something like that instrument with an autonomy that can tell you, hey, at time 32 in this portion of the image, it looks like there's something swimming around. Please check this out and see if this is what you're looking for. Same thing for like closing down beaches, right? If you want to know if there's harmful bacteria in the water, someone just goes out, scoops up a little water, goes back to a lab and runs through a few slides. Well, what if we could do that automatically? Like that would be huge. Um, and just to close that out, even our oceans here on Earth, we don't understand all the bacteria that are in our own oceans. I think, again, this is another place, don't quote me, but I think that I've heard like over 90% of the bacteria here on Earth, we don't fully understand. And so just putting maybe 10 of those on like container ships, and just having them, collect data for 2 years. All right, now you have, I don't know, 10 petabytes of data. It's probably going to be a lot more than that. But here's 50 recordings. The autonomy is telling you, like, you should look at these first. We've characterized them down into like 4 types of motility that we tend to see. Here's where you should spend your time looking first, rather than having to manually watch, all those videos, which at some point becomes impossible. So that's kind of why I'm excited about science autonomy. I think there's a lot of ways that we can help empower scientists to spend more time looking at interesting data and get good data back to Earth, rather than have them kind of manually flipping through, or just reducing the tedium that they have to deal with.
Right. And the work for all those grad students.
Yeah.
That's amazing. The applications are, yeah, pretty endless. I can see where this can be used all over the place. So given that, I think it's a good time actually to check in with Nikki to see what's going on out there. I'm sure we've got questions coming in. Nikki, how's it going out there?
Oh my goodness, the chat has been so very busy. Tons of questions for you, Marc. First off, tons of happy birthdays from the chat. So happy birthday from everyone in the chat as well. But one of the biggest things people are kind of wondering about on all the different channels is, how do you get into a field like this, and what kind of advice would you have for a college student, a grad student, a high school student who wants to do something like this?
Yeah, that's a great question. I think, like, hopefully from this picture and talking about the team, I think one of the things that's worth stressing is that there are a lot of really tough problems that we want to solve or look at, like trying to find life. And it takes so many different people with different backgrounds, different areas of expertise, like different ways of thinking, to be able to actually make progress on those problems. In some ways, like, to me, it almost feels freeing in a way, because the like, gone are the days that you could have a kind of like a single inventor make huge strides on some really important problem. You need to have these really big teams. And so I think what's nice about that is that means, like, you can kind of think about and try different areas within, like, learning different types of skills, or learning different types of knowledge, or studying different things, and those can all be relevant to working on some of these big problems. So I think my advice to, especially to the college students and high school students, try a lot of different things, do internships, shadow people, just do even informational interviews with people who you think they have interesting work. Ask them how their day to day looks, and then maybe try to understand, okay, like, what am I most passionate and excited about day to day, and how do I, like, what type of really big problems do I care about? Like, how do I connect those two aspects? And I'll say, spoiler alert, like, I'm still figuring it out too, right? It's probably, what are you passionate about is probably one of life's toughest challenges to try and answer for yourself. So it's always a work in progress.
Yeah, I mean, I always say the toughest things are the things most worth doing, but that's some great advice. You mentioned as well how difficult this is, this kind of observing and understanding and potentially looking for life. And we've had quite a few questions on that. So I'm actually going to put two together. Neil on Facebook wants to know, what happens if you actually find life with owls? And Bill on YouTube wants to know, if life is found on another place, is there a way to determine if it came from an asteroid or a comet strike on the early Earth?
Yeah. So the first question, how do we determine if we found life with owls? And at the end of the day, owls and the instruments and the autonomy I've been talking about are not going to replace the scientists. I think that is one of the most important bits to understand. We are just trying to send back the best data that we can. The science team will make the final conclusions that they can based on the data. And so they'll be working with, I mean, honestly, just the entire scientific community probably at that point, because this is such a huge problem. But they'll be putting together the evidence, and it is going to take an extraordinary amount of evidence, because this would be an extraordinary claim if we were to say we actually think we found life. But yeah, at the end of the day, that decision will go to the scientists to defend. The other question, yeah, about where does life, like, how can we tell if it came from somewhere, like if it was multiple places it originated, or did it originate in one place and kind of spread? OWLS isn't going to answer that per se, but I think there will be some interesting, like, as the data comes off owls, and maybe we did find something interesting, we'll be looking at some of those questions. So you could ask, one of the things we'll be looking for is nucleic acids. So that would be really interesting if we see that it seems like the cells, or whatever we found on the ocean of Enceladus, for example, also uses DNA. That's going to be really interesting because that's going to start prompting questions. Okay, well, maybe there was some kind of cross contamination, if you will, from like a meteorite strike, and those meteorites got transferred between multiple places in the solar system. That's possible. I think those theories are definitely out there, and very well thought about. But what if it doesn't contain DNA? What if it's like something completely different? That would be really exciting too. And I think looking at those individual biosignatures would probably help, start to direct our thinking about which of those hypotheses would be potentially correct.
Yeah, so I mean, there's a lot of different ways we have to think about this, but I mean, I love what you're sharing, and more people are asking more questions about life. And Alexis on LinkedIn wants to know, would amino acids still be the building blocks of life for something that doesn't have a carbon base?
Yeah, this is one of those, it kind of comes back to the fact that this is in some ways like a paradoxical search. Like, we understand our like life on Earth reasonably well. And in some ways we don't, like I mentioned at the start, we don't know exactly what we'll find somewhere else. And so it may not be carbon based. There may be some key differences from life that we understand on Earth. And so I think that's what comes back to that, the strategy of OWLS, right? We try to put as many instruments as we can on this life detection suite and look in as many independent ways as we can. So even if we don't see, for example, we didn't find any amino acids at all, there's a bunch of other molecules that we don't fully understand, but we took these microscopy videos and indeed there's something swimming around in there. Like, that's really interesting because we have ways to both look on the chemistry side, but we also have ways to look for signs of life that don't involve chemistry at all. And so I think that's the important bit about the strategy is, as you try to carry out this search and you realize how hard it is, you have to be able to try and count for as many of those scenarios as you can.
It's good that the scientists are thinking of all of the things out there so that we know that. But it is weird to think about life that could be non-carbon based. So we've got some other great questions. I think we've got time for maybe 3 quick answers. So Rob on LinkedIn wants to know, could owls be used on Mars at the ice caps? And Dee on LinkedIn also wants to know, could we go to water exoplanets? So I know we talked about Enceladus and Europa, but where else could these things be applicable? Where else could we use owls?
Yeah, Mars is definitely one that people are talking more about. I think we're still searching and trying to understand more about where water is within our solar system, and Mars is one that's popping up a lot more. So maybe at the poles, but I think I've heard people talk a lot more about lava tubes as well, like old lava tubes that could have liquid water underneath. That's the key, is we want to try to find places where there's liquid water present, and those are going to form some of those target locations that we want to send the OWL suite. And yeah, it would be really, really interesting to send it to some exoplanets even further out. I think we will be especially challenged then to make sure we can transmit the data back home, because the distances there, when you start talking about going between solar systems or, to other stars in the galaxy, we're going to be really pressed. So maybe that's a good way to confirm that I'll have job security is try to send these things even further out.
Job security is a great answer. I love that for you. I think we've got time for one last question, and you have inspired some people, but Anaya on YouTube wants even more information. So, Marc, how did you know that you wanted to do owls?
Oh, it's so hard. I don't know if I can even explain what it is. I think just in general, there are so many very cool and very tough challenges and problems in the scientific domain. And I think for me, I don't know, I felt a draw, especially to Mars and to some of these big challenges, and being able to work with such a big team. I think that's what I enjoy most about it. Um, it's a feeling, I guess. Again, I don't know if I can like write down what exactly got me to this point. But yeah, I'll go back to what I said earlier. If you if you're trying to figure out like, what exactly are my passions, try as many things as you can. You don't come out and know your favorite ice cream offhand. You need to try like 20 or 30 or 40 varieties over your lifetime to really hone in on what your favorite ice cream is. It's the same thing for doing work. You got to try a lot of different things and collect your own data in a way to see what it is that most inspires you and where you click.
Oh, that's fantastic. Those are great words. Nikki, those were fantastic questions. Thank you, all of you out there on social media, for your contributions tonight. So that really is just about all the time we have. So again, Marc, thank you very much. Again, happy birthday. Nikki, thank you
Thank
as always for your great job behind the scenes there, doing this whole social media question handling thing. I really appreciate that. All of you team behind the scenes, directing this and making all this happen, all of you handling the chats out there on the various social media platforms, thank you very much. And of course, all you viewers, thank you for tuning in. We couldn't do this without you. We wouldn't do this without you. So we really do appreciate your time and effort just to tune in and see what's going on with us. So please join us next month when we'll discuss what can be learned by studying those occasionally threatening near-Earth objects. That should be a fun one. Until then, my friends, stay safe, stay well, and stay curious. Have a great night.